So we've talked about the supporting cells or the neuroglia cells. So now we're going to talk about the main electrical cells of the nervous system, which are called neurons. And these neurons have three main parts to them. The area where the nucleus is found is what is called the cell body, the biosynthetic or receptive region. It's also called the soma. And so the soma or cell body, you need to remember that the word soma does not mean the same thing as somatic. So somatic typically refers to the entire body, not just one single neuron. So don't let that confuse you. The second part of the neuron is the receptive part of the neuron, which is called the dendrite. And the dendrites are short tapering receptive regions. They are points of synapses with other neurons, and they have electrical signals that are conveyed as graded potentials. And that's very important. They're also unmyelinated. So both axons and dendrites are extensions of the cytoplasm. So they are processes that are extending out of where the cell body is located at. So the axon is then it generates nerve impulses, which are called action potentials. So we're going to learn in this semester how action potentials are different than graded potentials. Axons are also outmyelinated, and they are responsible for outgoing signals, whereas the dendrites are responsible for incoming signals. So let's look at our diagram a little bit more closely here. We can see the nucleolus inside the nucleus, rough endoplasmic reticulum, which is called a nicel body. We then have the axon, which takes the signal away from the cell body, and those are called action potentials. I'm going to abbreviate them with AP. And notice at the very beginning of the axon, we have what's called the initial segment called the axon hillock. And the axon hillock is also called the trigger zone. It's called this because it has a high concentration of sodium channels. So the brackets here mean concentration. And then there is a synaptic cleft, which is going to be a space here between the axon terminal and the next neuron. So we'll talk about the axon terminal later. That's where a neurotransmitter is going to be released from. The Schwann cells are in the peripheral nervous system, produce the myelin. And again, that allows for these action potentials to leap from what are called node of Ranvier to node of Ranvier. So in this micrograph up here, we can see the neuron cell body, the soma, dendritic spines, and the axons sometimes are difficult to view, but this is probably the axon. But the other thing I want to point out is that the cell body also has no centrioles. So this means that there is no cell division. So this is why cell neurons cannot reproduce. The last thing on this slide is what's called the node of Ranvier, which I've already mentioned. And these are gaps between the myelin sheaths, and they allow for the leaping of action potentials from node to node. So I know there's a lot on this slide, but you can always, play at lower speed, pause it, take screenshots, go back and review it. So the neurons are the most important cells that are found in the nervous system. Our next slide is going to have a very important chart, and this chart is going to compare basically items that are in the central nervous system versus the peripheral nervous system. So when we talk about collections of cell bodies, we call them nuclei in the central nervous system. When we're talking about collections of neurons neuron cell bodies in the peripheral nervous system, we call them ganglia. When we talk about axons, bundles of axons in the central nervous system, they're called tracts. And in the peripheral nervous system, axons are simply called a nerve. So those are important terminologies that you should know. When we look at an axon specifically, these nerve fibers, there's a lot of important information about them. Because of this myelin sheath wrapping around the axon, we see the Schwann cell plasma membrane right here. The Schwann cell envelops the axon, rotates around the axon, wrapping around it. And the Schwann cytoplasm is forced between the membranes to surround the axon to form this myelin sheath. And what the beauty of this myelin sheath is that it allows action potentials to leap from node to node. So it comes from the word saltere, which means to leap from node of Ranvier to node of Ranvier for a very fast nerve conduction. So myelin is a whitish, fatty protein lipoid structure. So whenever you see white matter in the nervous system, it's because of myelin. So it protects the axon, electrically insulates it, and increases the speed of the nerve impulse transmission. So in the peripheral nervous system, it's formed by these Schwann cells, forming the myelin sheath. And then the membrane of the nucleus is called the neurolemma. So here we're kind of looking at the length of the axon. Here we're looking at a cross-section of the axon. And we see the myelin sheath surrounding it. So the difference here is the inside of the axon is negatively charged because the phospholipid bilayer works like a battery. The inside's negative, the outside is positive. So there's extracellular fluid outside of the cell, intracellular fluid inside the cell. Just like we learned, there's sodium on the outside, chlorine on the outside, and potassium and amino acids that are on the inside. But due to this insulation, an action potential doesn't have to form at that region of the axon. Instead, it's going to be able to leap from node to node. So myelin is very, very, very important. And we'll talk more about that coming up with the resting membrane potential.